Hydrothermal liquefaction, or HTL, is a process that changes the chemical properties of water. When water is heated to 250 to 350 degrees C and maintained at a pressure of 10 to 20 megapascals, it will break down macromolecules such as lignin and cellulose into smaller value-added products. An attractive attribute to this thermochemical conversion platform is its ability to process high moisture feedstocks. The process is ideal for waste biomass such as algae, manure, or sewage. So coupling algal wastewater treatment with HTL conversion of algae is an attractive system. However, this process will generate a large volume of a material called aqueous phase that must be considered for the system to be sustainable. This material is known to contain potential nutrients such as nitrogen or phosphorus. However, it is inhibitory to algal growth and its environmental impact is largely unknown. Therefore, characterization of this aqueous phase is necessary to determine its utility or treatment. So in our first attempt to characterize this material, we performed a two-stage liquid-liquid extraction. However, we found that our extraction efficiencies were very low, and the GCMS analysis of the extracts was hindered by the chemical complexity of the species present. So in order to increase our extraction efficiencies and to obtain cleaner GCMS data, we performed a sample fractionation method for dissolved organic matter. This method separates a sample into 4 fractions based on the polarity of the chemical forms present. Here is a mass balance for the fractions that we obtained, and you can see that the hydrophilic fraction contains the vast majority of potential nutrients. However, this fraction also contains material that can inhibit algal growth, such as sulfur and organic nitrogen. A toxicity test, a chronic toxicity test that we performed on the fractions utilized Chinese hamster ovary cells. This assay measures the cell reduction as a function of the concentration of fractions and is incubated for a period of 72 hours. The other toxicity assay that we employed was utilized bioluminescent bacteria, and like the chronic test, we assayed the organism over a varying concentration of fractions over a period of 5 minutes, and we looked at the reduction of light emitted. So here are the chronic and the acute toxicity tests, and you can see that both assays agree quite well with regards to the toxicity trends. The trend observed was the hydrophobic base fraction, the hydrophilic fraction, the hydrophobic neutral, and the hydrophobic acid go from most toxic to least toxic. So to characterize the hydrophobic portion of the sample, we enlisted help of colleagues at LECO Corporation in St. Joseph, Michigan. They performed gas chromatography, time of flight, mass spectroscopy on the extracts that we obtained. And what we got is the base fraction alone contained 800 structures were identified in this fraction, and the vast majority of them were nitrogen-containing heterocyclic compounds, most likely due from the degradation of proteins present in the algae. The major classes of compounds identified were pyridines, imidazoles, pyrazines, amines, and pyridinamines. And most likely the toxicity of this fraction is due to many of these compounds or their interactions. Now, the hydrophobic acid and the hydrophobic base fractions were much less complex. The hydrophobic neutral and the hydrophobic acid fractions were much less complex than the base fractions. However, here as well, they're dominated by nitrogen-containing compounds. So, the hydrophilic fraction contains the elements required for algae growth, and this fraction will be the focus for nutrient recycling. However, our toxicity data indicates that these fractions may also inhibit the growth of algae. Since the hydrophobic fractions do not contain elements that are useful for nutrient recycling, this material should be removed. Now that we know this composition of the hydrophobic part of the sample, we can now target and identify treatment technologies and methods to remove this portion. To acquire a deeper understanding of the hydrophilic fraction, we are planning to fractionate this sample into acid, neutral, and base components as well. Then we will perform characterization and toxicity testing of these portions to get a deeper understanding of this material. So the data obtained and the methods developed for this project will play an important role in the treatment and use of this material. In addition, we can use these methods to observe changes in the HTL materials as the feedstocks change and as the operating conditions are explored. Other possible applications of these methods can include anywhere where there's complex samples produced from energy-water nexuses, such as hydraulic fracturing fluids, pyrolysis waters, or retort waters. So, in conclusion, ISTC possesses the expertise and the facilities to analyze complex samples. We are ready to use these methods and many others to support projects within the Prairie Research Institute, the University of Illinois, the state of Illinois, and far beyond.